All right, that looks better. All right. Hi everyone and welcome. My name is Nicholas Yu and welcome to tonight's presentation on Wild Orchid Watch. Just a reminder that this presentation is being recorded, and on behalf of Willoughby City Council, I would like to acknowledge the traditional inhabitants of the land on which we stand, the Aboriginal people, their spirits and ancestors. We acknowledge the vital contribution the Indigenous people and cultures have made and still make to the nation that we share, Australia. So, Wild Orchid Watch, it's a national citizen science project designed to collect, record and share scientific information about Australian native orchids. And tonight we have two guest speakers. Our first guest speaker is Greg Steenbeeke. He's a vegetation ecologist and has worked in the environmental industry for over 30 years. He's also a member of the Australasian Native Orchid Society. Greg will be talking a bit about what orchids are, their ecology, anatomy, some of the orchids you might even find on your bushwalks, and the importance of why we need to be collecting data on our native orchids. And this will tie in with our second guest speaker, Rosalie Lawrence. So Rosalie Lawrence is an orchid educator for Wild Orchid Watch. So she'll be talking about the project itself, how you can sign up and do your and collect your sighting, your data, and the ethics behind collecting this data as well. So we hope you enjoy tonight's talk. And if you have any questions, please put them in the chat box, which is down at the bottom of the screen and roughly in the middle. And yeah, I think we'll get the ball rolling. So I'll put it over to you, Gregory. Let's see, turn off.
You should now be able to see my screen, I'm hoping. Can you just confirm, please, Rosalie or Nick, that you're seeing the what is that orchid front page?
Yep,
Yes.
I can see what is that orchid, yes.
Okay, are you seeing only that or are you seeing other information as
No,
well?
only that, Greg.
Just
Okay, fantastic. All righty, so as Nick said, I've been in this game pretty much all my life. I'm a third generation orchid grower, but I've also trained as a botanist through Sydney University and have worked across much of New South Wales, being based at various places around the state during 30 years with the Department of now Planning and Environment and all its 14 or 18 previous iterations that I was with it with. For the last 14 months, so I've been doing my own business, working in environmental consultancy. So when it comes to orchids, we want to know what it is. You know, I'm going to start, as I said, with my take-home message. And the reason for that is I don't know how many of you are going to fall asleep through the talk, but let's look at it this way. The beginning of wisdom is to call a thing by its right name. When it comes to using names, it is very, very important that you become familiar with the concept of nomenclature, the way in which plants are named and animals are named. And the reason for that is because when we're talking about a species, we're talking about something that is defined clearly. So in this instance, when it comes to talking about a common item, and we're all hopefully talking about the same thing, using the right name means at least we're in the first step to getting there. So as I said, we call this nomenclature. And in nomenclature, we have a classification hierarchy going from kingdom through phylum, class, order, family, genus, and finally the species epithet. So in that instance, you can see there's the full description for the Sydney rock orchid, Dendrobium speciosum. I try and avoid using common names where I can because it's much clearer if I use the scientific binomial rather than to talk about Sydney rock orchid or king orchid or any of a dozen other common names that it's known by, and none of which are those used by the local inhabitants that were first here. So how do we know where orchids fit in the grand scheme of things? Well, here's the family tree, if you like, of all of the higher plants. So these are all the plants that have vascular bundles or a vascular system. So those range from ferns through to the higher flowering plants. Where do orchids sit? They sit basically there in the asparagales. So they're relative to things like asparagus and so on. And as you can see there, we've got the phylum, which is the vascular phytae. We've got class, which is Monocotyledonae. And then we've got the order, which is Asparagales. So within the order, you can see here are some of the other families to which orchids are related, and you'll notice that they are basal, or the most primitive of the Asparagales. And it's an interesting concept to talk about things as being primitive and advanced, and yet at the same time, everything is equally evolved. So when we look at where orchids fit into the family orchid, the order Asparagales, they fit at the very base of it, in that they have the basics description of being in the Asparagales, but they don't comfortably sit with the rest of everything else that's in that order. So to allow for things like that, we can use alternative additional levels of classification. So within the family Orchidaceae, for instance, we can have a subfamily Epidendroideae, then a tribe Podiciliae, Dendrobiinae, back to the genus Dendrobium. So we've inserted 3 separate layers of clustering. Each one clusters more than the one below into that gap between the family and the genus. We can then go below the genus and do subgenus, section, species, and even then beyond the species down into options like subspecies, variety, forma. So within Orchidaceae, we have 5 separate subfamilies. Of these, 4 occur in Australia, which are the Apostasioideae, Phenelloideae, Orchidoideae, and Epidendroideae. We don't have Cypripedioideae, which are the slipper orchids of Asia and the Americas, but we have everything else. And when we look at the arrangement of these families, it's like the family tree again. You can see we go from the most primitive, which is Apostasioideae with its 2 genera, through to Epidendroideae and Orchidoideae, which are the most, what we call, specialized of those subfamilies within the orchids. And we call them specialized because they've taken on things like selective pollination by particular individual species and things like that, which lead to high specificity, I'll get there, high specificity of the relationships that they show to other organisms. So what are orchids? Well, orchids are the largest family of flowering plants. There's more than 25,000 species, and depending on who you talk to, an awful lot more. And the other large families, Asteraceae and Poaceae, all have about 20,000 or 22,000 species. They are distinctive. They're typically insect pollinated. There is actually an orchid that's pollinated by a grasshopper, but anyway, and highly specialized with regard to the habitat they occupy and the interdependence they show to other organisms. So there's the technical definition of what an orchid is. This is from the MOBOT, Missouri Botanic Gardens website. So if you wanted to define an orchid, there's the text there. But really, let's look at what it means if we look at it more simplistically. It is a herb that is a non-woody plant, which is dependent on fungi, mycorrhiza, especially at germination. So they're mycoheterotrophic at germination. They rely on nutrition coming from the fungus to stimulate the growth of the seed. Their roots typically lack hairs. Cypripedioideae and some Orchidoideae do have root hairs, but generally orchids do not have root hairs. They have racemes, which are a single axis of flowers, which each individual flower is on a stalk. The flowers have a single axis of symmetry, which goes from the top of the flower through the middle to the bottom of the flower. It doesn't have a radial symmetry, so you can't choose any plane on which to align an orchid. They always have top to bottom symmetry. The flowers are usually inverted, or what's called resupinate. If you look at the stalk of a flower of an orchid, you usually see there's a slight twist in that flower stem. The sepals, the tepals, which is a general term for sepals and petals, are free. That is, they don't join to each other in most cases. There are some specialized options that we'll look at in a little while. one of the inner whorl, so one of the petals, if you like, forms a labellum, which is a specialized structure that permits it to get pollinated. The stamens and styles are usually fused, with very few exceptions, and there's often only one anther developed, although the primitive families, like Apostasioideae, has 3, and Cypripedioideae has 2. And the stigma is wet, that is, it receives the pollen by being allowing it to adhere to it as a sticky. It is sticky. The ovary is inferior, that is, the place where the seeds form in the flower is below the flower itself. So it's between the tepals and the stalk of the flower. The seeds are minute. It's not uncommon to have 1,500 plus seeds per fruit or per flower. And in some orchids, like some of the Asian cymbidiums, well over 1.5 million seeds has been counted in a seed pod. So when you talk about a 1 in a million chance for a variation to occur in a species, it happens pretty much every pollination in Orchidaceae. So let's look at a couple of these subfamilies. So Orchidoideae, which is found, as you can see there, across much of the globe. They are terrestrial herbs with root tubers. Occasionally they are growing on trees in moss beds and things like that. It's a bit hard then to call them an epiphyte, but because they're still growing in a soil or a humus that's actually up in the tree. They may be secondarily epiphytic. They have a global distribution. They have mealy pollen, so that means that the pollen itself, even though it's clustered together, is quite granular. And then the Epidendroideae, which is the other major subfamily, is the most abundant and diverse of the orchid subfamilies. It has pretty much the same distribution, being global, as Orchidoideae. The majority of Epidendroideae are epiphytes or epiliths or lithophytes. So they're growing on things. They're not necessarily growing in the soil. And when they're in the soil, usually they're in a humus-rich leaf mold type situation. So they're not rooted into what we would call the mineral soil. The pollen is in waxy clusters. So when the pollen occurs in flower, it's actually in hard clusters. And if there are storage organs present, then they're usually the stems or succulent leaves, not the roots. Dipodium is one of the few exceptions to that. The succulent root is the standard storage organ in the Orchidoideae. So coming back to nomenclature, which is the method of presenting information in the form of names. 2 different published and more or less standardized codes exist when it comes to how we name plants. There's the scientific naming system, the ICBN, or the International Convention for Botanical Nomenclature. And then there's the horticultural code, or the International Code for the Naming of Cultivated Plants, or Hort Code as it's also known. Getting how we name things right is vital. It's critical for how we share the information and what it is we're sharing information about. When we talk about flower plants and flower parts and plant parts, we're talking about, again, another specialized set of terms. So we've got tubers in the ground orchids. We have a sheath or a tunic, which is found pretty much in the standard in Caledonia. We have roots, as I mentioned before, rarely with root hairs. What looks like root hairs in many orchid roots is actually the mycorrhiza of the fungi that are infecting the roots, spreading out through the mineral soil adjacent. There's the stem, which in most Orchidoideae is ridiculously short. There's the petiole, which is the stalk of the leaf blade itself, which is the green or otherwise coloured broad part of the leaf that's photosynthetic. And then we have the peduncle or scape of the inflorescence. We can have a corm-like pseudobulb, in this case of the Epidendroid species Geodorum. You can see there it's sticking out just above the soil surface. We can have a leaf rosette, where we have all the leaves produced very close to each other on the stem. Not quite a whorl, but very close to it in appearance. And in things like Dendrobium, we can see here we've got the leaf blade, as I said, the broad flat part of the leaf. The pseudobulb, which is the swollen stem. The roots. And in another related group, the bulbophyllums, we can see again, we've got another thing here called a rhizome, which joins between the pseudobulbs. We have the leaf petiole, which is quite well developed in many bulbophyllums, and then the leaf blade. The peduncle, as I mentioned before, is the stem or the scape of the inflorescence. You can have the pedicel, which is the stalk of the flower, and then the flower itself. Plant and flower parts, when it comes to how we describe inflorescences, we can have a solitary flower, like most of the pterostylis and many of the caladienias. We can have a raceme, where all the flowers are arranged on a common single axis, or we can have a panicle, such as we see here in Acreopsis. Panicles are very rare in orchids. It's a branch of racemes, a many branching cluster of racemes. And in all of them, we have a flower, we have bracts, we have the peduncle. So we have common characteristics shown right through. These parts of the flower are very important when it comes to how you get around to identifying what these species are, because you need to know the names of these parts. In most botanical texts, such as the Flora of New South Wales Online, or PlantNet as it's otherwise known, the glossary is there as a hyperlinked list, pictures and descriptions. And if you don't know what a term means, always go to the glossary. That's why it's put in. If we look at this flower of Caladienia, we can see we have the dorsal sepal, which is behind the column. We have the lateral sepals, which are, if you like, the legs or the front of the flower. We have the petals on either side. A modified petal called the labellum or lip. We have the column itself, which is the fused structure of the style and stigmas. Style and anther, sorry. We have the anther cap, which is the cap that sits on top of the pollen. The column wing, which is just an outgrowth from the side of the column and restricts the way in which an insect pollinator can move, or in this case, probably also provides a little bit more advertising for that broad red inside of this Caladienia. Labellum side lobes and labellum mid lobes are important parts of that part of the flower. And then on the labellum itself, you often get callus or calli, which are in rows or individual and often distinctive in shape, color, count. The features generally tend to be consistent throughout orchids and quite easy to find once you get familiar with the shape of the flower. So we have the anther, the stigma, which, as I mentioned earlier, is sticky, the petal, the labellum, the lateral sepals. Again, if we look at this Chyloglottis, we can see here much the same structures, but in this one, we've also got part of the sepals modified into what's called an osmophore or a smell gland, which is what osmophore basically means. You can see there clearly the ovary, which is where the seeds form, and how it's inferior, it's below the flower, between the flower and its stalk. You can have a nectary spur on some species. They're very rare in Australian orchids, but we do have them. And also too, in plants like Dendrobium and Bulbophyllum, we have what's called a mentum, which is an extension of the base of the flower from which the labellum hinges. Now, I did mention most orchids have free segments. One of the main groups that we get around Sydney, Pterostylis, has fused portions of the flower. So the lateral sepals are fused together, and the dorsal and the petals are also fused together into what we call a galea, or helmet, if you like, which is what galea means in ancient Greek. We have the anther, again, quite obvious with its yellow pollen. We have wings on the column, and that's where Pterostylis gets its name from. We have the labellum itself. We have a sinus in some of the species, which is that little gap in the middle there of the two fused sepals. The sepals there are fused, and the front portion is called the boss. You can see there the lateral sepals on both of these flowers. The one on the left is Pterostylis curta, the one on the right is Pterostylis longifolia. This is Pterostylis, so let's have a look at some of the species of orchids we're likely to see. Not so much around Willoughby. There are many of these that will be occurring around Willoughby and sort of the north shore of Sydney, but as a person who grew up in the Blue Mountains and Randwick, it's an interesting combination. My father was at UNSW. I've got a familiarity right across the Sydney basin. I'm now based in southwest Sydney. So this is Pterostylis newton, the nodding greenhood, which is very common around Sydney, typically flowers in wintertime. Pterostylis hispidula, which often flowers a little bit before it, is distinguished from nutans by being a smaller flower, and the hispid that gives its name to hispidula is a roughness of the galea of the flower. Pterostylis curta, which is another one of these colony-forming pterostylis, immediately distinguishable by this twist in the labellum. Pterostylis baptistii, the largest of our greenhoods, those flowers can be 6 or 7 centimetres from top to bottom. It is really quite an impressive plant. Favours wet sclerophyll forests and edges of rainforest. Pterostylis acuminata is the first of our pterostylis that we see flowering around Sydney, typically in flower around April, also known as the autumn greenhood. Pterostylis pedunculata, the maroon hood, is a very dark flowered species. You'll see it in places in Royal National Park, there are some, but also too up around the Upper Blue Mountains, there's quite a few there. Pterostylis consina, one of the fork-tongued greenhoods. You can see here the labellum actually has a fork in it, so sometimes it's called the snake-tongued greenhood, although that name is preferentially used for this one, Pterostylis ophioglossa meaning snake tongue. Again, it has a forked tongue. Pterostylis longifolia is one of the multi-flowered pterostylis and very common around Sydney. Then we look at another genus. This is Kyloglottis, or the ant orchids, Kyloglottis reflexa, Kyloglottis trapeziformis. The plants that you're seeing in cultivation either come from my collection or from the collection of others that have been cultivating these plants. Sometimes I just don't have a picture of the plant in the wild. Kyloglottis formicifera. Formicifera means ant-shaped, and you can see there the little ant on the labellum with its eyes, its body, its legs, and its abdomen. These are sexually deceptive pollinators. They convince a flying ant to come and try and mate with the flower, and as it tries to fly off with this fake female ant, it lifts the labellum and bangs against the anther, knocks the pollen, which has got a sticky disc, onto its back, and then when it tries it again with a different flower, it flies up and the pollen is pushed against the stigma, which is sitting right behind the anther. Kyloglottis semi-nuda, meaning partially naked, and you can see here the labellum has this naked tip. Kyloglottis trilabra and Kyloglottis sylvestris. So as you can see, there's quite a diversity of these species. They all look roughly similar, which is why when I mentioned before, it's good to know what the names are for each of these characteristic features, like the osmophores in sylvestris. It means that you can then use the keys in places like PlantNet to chase up that information. Kyloglottis diphylla is rounding this group out. Microtis is another one that we see very commonly around Sydney, often in just wet lawns. It's a colonizer species. It will actually quite readily adapt itself to any sunny, bright, grassy spot, especially if it's a little bit damp. Tiny green flowers, but as you can see, still structured like an orchid. They have the lateral sepals, the labellum, the petals, and a broad dorsal. Two species are common around Sydney, Microtis unifolia, which was the previous one, and Microtis parviflora, this one, which is distinguished by the shape of the labellum being more elongate. There are a couple of other species around, but I don't have photos of them. Prassophyllum looks similar to Microtis, but the flowers are upside down. Remember, we were talking about resupinate or non-resupinate. In this species, it's Pterostylis flavum, has the labellum uppermost. Genoplesium is related to the Prassophyllums. They have larger flowers than Microtis, but generally smaller than Prassophyllums. This is Genoplesium superbum from two places it's known, Western Blue Mountains and down near Maroon on the Southern Highlands. It's an endangered species. Salamitra, the sun orchids, which were out beautifully on the weekend up around the Upper Blue Mountains, with species absolutely enjoying the bright, sunny, warm days of spring. And this group, Phalaenopsis, typically will only see in flower in summer or spring on those bright sunny days.
Sorry, Greg, I'm sorry to interrupt. We're sort of sitting on the 7 o'clock mark now.
Okay.
Yeah, we might let Rosalie do her part of the talk, and if we have time, we'll jump back into your bit.
Yeah, that's all right. I'll just race through.
Yeah, sure, all good.
So
Yep.
you see there more diversity of orchids. Coming through the ground orchids, up into some of the tree orchids. And yes, all of these orchids are in the Greater Sydney area. All right, so when it comes to the importance of information, the quality of the observation and the reliability of the identification are critical parts. The accuracy of the location is good when it comes to trying to distinguish species. The potential for communication is enhanced by the use of accurate nomenclature. And that leads us into how WOW, Wild Orchid Watch, becomes an important facet in what we're trying to portray here. So WOW allows us to do threat management, cover the taxonomy, look at the phenology, how and when things do their life actions, such as flowering or resprouting after summer. Allows us to look at restoration of populations or even reintroduction to where populations have been lost, and allows for longitudinal study over time of how things change and provide spatial data to those with access to it. So, Rosalie, now you're going to show us what the WOW interface looks like.
I am. As soon as I get to screen share.
Well, I've stopped, so it's all yours now.
Okay. Right. Share. That come up on your screen nicely, everyone?
It has.
I can hear that. Yes, they did.
Good.
Rabbit orchid.
Yeah. Common names, even though Greg has just said about common names, they still get used around the place. So that's just one of the things. So I'm going to talk about the WOW app, but just very, very quickly, one of the, sorry, I'm just moving something about so I can look at some notes. With WOW is that, as was said before, it is a national project. It's actually linked to iNaturalist. So if anyone has used iNaturalist, or rather, if you are signing into WOW, you actually have to sign into iNaturalist, but the app runs separately from it because there are features within the app that do not exist on iNaturalist. iNaturalist primarily, I'll just briefly talk about iNaturalist and then I will move into the app, is about opportunistic observations. So you're walking along, you see something, you snap a picture and you put it up. But WOW itself is more structured in that we have a series of questions that we ask. There's a series of photos that we want as well. And this is to help us get a more rigorous data than just the opportunistic data. So I'll talk about these things as I go along. And I'll also give some hints. So I'm thinking, Nick, some of my notes I can actually give to you later that can be shared out because I'm quite happy with that. So we'll actually talk to the app. This app, you can download it onto your computer as well as onto your phone. And so I'm using my computer to do it. So both the interface looks the same, very similar on your phone as what you're seeing here now on the computer screen. My name on iNats is Orchid Rose. I'm based in South Australia, by the way. And one of the things is in South Australia, we don't have as many orchids as you guys have, and we definitely do not have the epiphytic and lithophytic orchids, which to a certain extent makes it a little bit easier for us to concentrate on the ones that we do have. So just briefly, the app is linked to the Wild Orchid Watch website, and I will have information on that later. There's a whole series of frequently asked questions. So I'm not going to go through all those questions, but some of them will come up, I'm sure. And the most important, or two important things I would point out is what we call the help button. I have a friend who's saying is when all else fails, read the instructions. Well, we put the instructions of what we're looking for, what our definitions are, what the questions are asking, there with you on the app so you can look at it while you're doing your observations. And so it's quite a lot of different, as I'm just scrolling through, you can see that the different ones, but I'll get to them specifically because what I'm planning to do tonight is actually do an actual observation. So you can see it in action. The other is this app has only been going for a short time and we are still really in the very early days. And so if you do have any problems, there is a report of problems section here that you can get an email to us and we can look at it because we do want to make this as good as we can. Now, the advantage about the app, one about these apps is Greg was going through and showing all the different features of the orchids, and you're not going to remember most of it. But with these apps, you actually don't need to know much at all. In fact, we don't mind if someone ends up putting an observation up that's not an orchid. It's better to give it a try than not at all. So I'm going to go into the observation. Let's get back up here. So you'll see a button down the bottom that is to add a new observation. I'm not sure if I'm going to see any questions, but if there are any questions as I'm going through the app, perhaps Nick, can you just jump in with the question for me? So to do an observation, one of the things we've done with setting up this observation is that we've got two modes. We've got here a detailed mode, which is the preferred mode we want to have for taking observations. But if you don't have the time, you can go, or you're still trying to learn it, you can go to basic mode. Now I've just put that into detailed mode because I'm going to go through the detailed mode. So there's the required information. There's also the help button to tell you what we mean by those pieces of information. So first up, we want a photograph of the whole plant. Now you can do this a couple of different ways. You can actually take the picture in the app, or you can take it and add it afterwards. It doesn't really matter. It's what your preference is. So when I was out on Sunday with the BioBlitz, I didn't have time to really do WOW observations because the issue with WOW is that it does take a little bit more time. It takes a little bit more planning to do. It's not the type of thing you do when you're leading a field trip with a whole lot of people. So because I know the questions, I snapped some things, made a mental note of what I was looking at, and I'm very familiar with the site, which was also helpful. So first up, you want a whole plant picture, and you just, you can drag and add that. Then there's habitat photograph that's required. So you can drag and put that in, and the same, let's do that one. Now there's some other pictures, and the thing with orchids, and Nick, I'll send you I'll put a slide up with this at the end that shows, orchids, you do need to photograph different parts. And each of the genera is often just a little bit different on the type of things that you need to show, that we need to see. So there is an article on how to photograph for orchid identification that I will pass on to Nick as well. So in this instance, this is why we've got this section of extra photos, because different features will give us different pieces of information. And I'm going to put in a whole plant. So I'm actually going to add two pictures for here. So here's a close-up of this orchid, which I will get to the name a bit later on. But the other is scale is very important. Your hands are good for scales, but we actually have produced some scale cards. And so here it is, I'm using the scale card to help not only with the size, but also colors. So sometimes when you're out in the field and you're taking photographs, it's very easy to get washed out pictures. With the scale card and the colors there, you can actually tweak your photograph to bring it back up to the color it should be. When you also do the scale cards, as far as measurements go, they need to be in the same plane. So in this instance, I've actually got that up close. So you can see the size of the flower. So when you look at this, the whole picture, this is cut off some of the picture, but in actual fact, when you're looking at the observations, when they go up, you'll see the rest of the scale card with the ruler measurements on it. So this one does not have leaves. If you see any floral visitors, yes, photograph them and put them on if you can, because that's all helpful information. The same as particularly for you guys in Sydney, photograph the host tree that your epiphytes are on. Now, the next section, which is anything here is required, we need to put in the species name. Now, if you don't know the species name, that's not a problem. You don't have to have a lot of orchid knowledge to contribute to the project. So you can just put in orchid, because someone will come along later and they will do the identification. So in this instance, I do know what it is. It's Grassodia. And it starts bringing up some suggestions. Oh, wait, I think I saw it just past, yeah. Sesamoides. And that's the potato, well, here we call it the potato orchid, but it gets called different names. The other is cinnamon bells and a few other names. But that name is Matt on iNaturalist, so we can keep it. Now, this is an important part. So your phone, reason for using the phone apps is because they do have GPS, but not everybody has the GPS turned on. I was helping a lady on Sunday who does not have GPS on her phone. For some reason, she can't get it functioning. But you have other choices. So if I had been adding this where I was down in the forest, then I would have clicked use the location. But I don't. The other is, if I know what it is, sometimes I go out and I'm GPSing sites, I might enter the GPS. But in this instance, we can manually input by dragging on a map. And I will get this to the right spot. Just bear with me as I come down. It should be, oops. You can also make these changes in iNaturalist as well. This is a little clunky, but we're trying to improve on some of these things. And I know exactly where I was, so I can pinpoint. So that's the location. Now I can check that on a map to see whether I've done that properly. And yes, I was at Chukaloo Campgrounds. So that's it. The other is the phone, the date and time, and that was found on the image, so that was good. Otherwise, I can enter it manually. Now we come to the type of orchid. So you've got a drop-down menu, and you can select, well, this is a terrestrial, and then we come to a series of questions. which not all of them are required, but they are helpful. So yes, the landform. Now, there's a lot of different definitions here. And again, you can go to here and you have your landforms. Now, this was, yeah, this was an open depression. So let's get back. Landform, so go on here. So if I'm unfamiliar with the definition, that's not a problem. I can go and find out because it would do this. Soil structured, I didn't collect that, so I won't blame. Were there any rocks? This is where the rocks are within a metre of the orchid, and there were none. Now, if there were some, and it was a range of them, this is a multiple selection. So, for instance, I'll just come out of that one. You know, for it, if it had been something like that, you can put on more than one. The other is if you're doing lots of observations in one area, you can, to make it a bit quicker, if you're observing within a, say, a 100 square metre, that's 10 by 10 metre area, a lot of these observations aren't going to change. So you can just say select the last value, and that will help. Now, accuracy of population, unless you're actually doing a survey where your aim is to count every single flower, which we did a few weeks back, or whether you have an area that is within a grid, you're either going to do a partial count, as in you are not going to count all of them, or you're going to do an estimate type count. So I did do an estimate, and there were so many that was really good because I could just, it was an easy number to remember. So how many did you observe? Then search effort. This is not your opportunistic searching. So it's, if you've set out specifically to look for a specific orchid and you're spending time searching for it. So earlier this year, we spent hours actually looking for one particular orchid. It had been known to have occurred in the site, hadn't been seen for 10 years. So that's a long time to search. So this is only if you've gone in there with a specific task in mind. Where was I? So you don't need to answer that one. Accuracy of the search. Again, if you haven't actually measured out your area, it's always going to be an estimate. And the thing about estimating how big an area you've searched, we'll actually get onto that here. So your sorry. We searched an 8 by, no, it was a bit bigger than that. 12 by 2 metre size. So roughly 24 square metres. Now, how do you work out what size it is if you're not out there with a tape measure? Well, it's simple. We all carry a ruler around with us, and that is our size. If you know that you're 155 or 158 centimetres, your hand width from finger to finger across is that same measurement. Or if you know how big you can step out your stride. So it's an estimate. It's just to get an idea. And this particular population size. So the search area tends to always be bigger than the area you find them in. And this one was over quite a large range. So that gave the search. Now, the next series of questions is to do with the orchids themselves, and depending upon whether you've got a colony group or a single flowering. So in this, most of them were in bud. There were some in flowering, but there were no sinense flowers, no fruit developing, anything like that. And then, well, what was dominant? well, in this instance, it was the budding was dominant. There were not a lot of flowers out. Then you can have a look and see, was there any damage? Plants get damaged. They get damaged by grazing, they get damaged by insects, thrip, those type of things. Surprisingly not. Further on, we found more of that. And the other is, did you see any pollinators there? There are all types of pollinators in Australia. These are the main ones. And if you don't know what the insect you're looking at, you've always got the unknown. But in this instance, with the rains and everything else, yeah, there wasn't a lot of insect activity until later in the day. Now, this is where we start collecting information that may not necessarily be on the photograph. So what was the dominant vegetation around the plants? Well, it was actually, we were in tree area. And that, again, those terms are fairly simple. You don't have to be able to identify which types. So it was trees, was probably the main ones there, but it just gives you a bit of an idea. What was the height of the dominant growth? Again, it is but an estimate, but it's really done. So 0.5 meter. Most trees fall about for South Australia. We don't have overly tall trees, so 3 to 10 meters. And the foliage cover. So one of the things is, if you want to, you can take a photograph and do the canopy cover. And generally what I do when I do that is, when I'm taking the micro habitat on my phone, I just switch to a selfie one because the phone's looking straight up, and I take whatever's above. That helps to give that. But in this instance, the most dominant of the vegetation layers, it was not very shady. So then you've got the immediate land use, and this was in a conservation natural environment area, but the wider land use, we were actually in forestry SA lands, so that is production from dry land, agriculturals and plantations. Then any disturbances. This happens to be a lot of weed invasion. It's actually a campsite nearby. Yeah, there is some grazing that goes on as well. And yeah, so you just fill in those things. And then anything that you think's noteworthy. This is not kept. This is for your personal notes. Up here is if there's any other notes that you want to make. If you feel that the questions haven't covered something, you've observed something, maybe this is not a typical population, you can put comments in that it's anything you think could be helpful information. So having done all that, we're now ready to click save. So that is how you do the observation. And as there you are, you can see it's all there. And you can see the questions been answered. So let's go back to the home page. Now, what I want to do briefly is I'm going to switch to another screen, and it probably won't have come up in iNats, but I do want to show you how it looks in iNaturalist. You know, the one thing I forgot to do is work out how to share. Right. So what we're looking at now, can you tell me, can you just see, so this is where Wild Orchid Watch sits within iNaturalist. We don't recommend going through iNaturalist to put your observations in, mainly because it's easy to make mistakes with some of the questions, and you don't always get the required questions. We have got that available for historical records to go in, but that's fine. So yes, so that's what the page, the project looks like. And species, I'm going to go to species, identify observations. So from here, what happens with your observations is that they turn up in iNaturalist, and this is where the community helps with identifying orchids. Just trying to think of one to pick. Some people get optimistic with some of their observations. Let's have a look at this one. This observation, I can tell immediately, did not go through the WOW app because it's not obscured. And this is one of the things that's really very important, and we're trying to get the message across to people, is iNaturalist by default does not obscure any of the observations. But WOW does, because we are very much aware of sensitivity of sites, and we don't want people going to sensitive sites. So when a site is obscured, you cannot see it at all. I will come to that in a moment. So what would happen is, oh boy, you can just see, yeah, so people are learning now to take a lot more pictures, and that one I will agree is, so I can come along and agree. Now, this brings it up to research grade when it reaches 2/3 majority. If someone disagreed with me, they can come along, disagree with it, and that'll put it back up to genus level. So it actually surprisingly proves to be quite robust. Nick, I see that the time is getting on a bit here.
Yeah, I was just about to jump in.
I've covered all the things, but perhaps if I could just quickly share the screen, which shows how it is obscured, so that when people are looking, I have your observation up from the other day. And if we just show that picture, how did I do that last time? Do you know, I can't remember how I swapped that over to another screen. I'll stop the share and just come in again. Is that, can I do that? Yeah. Oh, found it. Because I think this is an important one to show. So this is Nick's observation, which he did some time ago. And I'll just come up to full screen. Now, I understand, Nick, the original observation is somewhere down here, is it?
Yeah, it was for Wildflower Gardens. Yeah.
Yeah. So this is what WOW does. It obscures the geolocation to 1 degree. Now, for us here in Adelaide, and it's probably going to be similar for you guys because you've got a similar latitude, is that is a 20 square kilometre. So for someone to go finding that, and I have noticed that 20 square, where it's obscured at, which I gather some down here, sometimes the obscuring might be they've just left it just in, but it's far bigger than that. So it is.
Rosalie,
Well,
the obscuring is to 0.1 degree, not 1 degree, because
Oh.
one is 120 kilometres across on each side.
Thank you, Greg. The point is, it's very hard to search a 20K area. I'll leave it at that. There's probably a lot more things, but I could send you a bit of a synopsis, Nick, that can go out, that
Sure.
you can share.
So it looks well, we got at least one question, so we know the chat function's working. Yeah, look, yeah, so many orchids, so little time. We'll probably have to, if we ever run this again, we'll probably might make it an hour and a half instead of an hour, I think. I was really captivated by all the species Greg was mentioning. So does anyone have any questions about the about orchids or Wild Orchid Watch?
I'll answer this question, Nick.
Sure.
Yes, so the WOW information also goes into the atlas as well, because it is going through iNaturalist, and it goes in obscured. If researchers are requiring the information, there is a procedure that, because we are linked with Adelaide University, there is a procedure of application for researchers to have access to that information. And we currently have 1 or 2 people that are using the data from WOW already, although we really need a lot more data in there for it to be really good, but they have found that it's even useful at this early stage.
Okay. I'm not sure what happens after the time, whether it cuts off or not. So, okay. So does before, oh, well, we got another question. Oh, it's a response. Amazing. Yeah, wonderful. Yeah, amazing number of species, so much to learn, and yeah, wonderful. Thank you. Oh.
So
Hey.
I'll give you some links, and if people want those scale cards, they just have to contact me on Wild Orchid Watch. Yeah.
All right.
With regard to the orchids, there are a number of very good handbooks and information sources around. There's a recently published book from CSIRO, the Guide to the Orchids of New South Wales and ACT, done by Lachlan Copeland and Gary Backhouse. Lachlan's from Coffs Harbour, Gary's from Melbourne. Both very renowned orchid people. It's, for the price of the volume, it is well worth it, and not overly heavy to carry around, but you'll see in there just the sheer numbers. And the best thing about it is, you know whether you're in the right sort of area because each species has a map.
The only thing I'd say to all of that, Greg, we have photographed and have an observation record of an orchid that is 400 k's out of its known range.
Oh, that's
what WOW will pick up.
That's nothing. That's what happened with one of the orchids on my farm up near Casino. The only locations known for it are Kingaroy and my farm, and that's 400 kilometres apart, and it's still not yet formally named.
Oh, this is a named one. But what I would say, yes, there are lots of books. So just one thing, when you're using both iNaturalist and WOW, is that they, for their naming convention, they are using Plants of the World Online, and that does not always agree with a lot of these authors.
Yes.
I'm looking into how we can best map the names that are being used. So, but I haven't yet come up to
and as you
solution.
said, Rosalie, as you said too, if you're not sure what the name is, you don't have to be exact.
Correct.
Come up through the nomenclature series to where you're comfortable, even if that's orchid, and there will be people like Rosalie and myself will look at these photos, and if you're lucky, we'll put a species name on it. And if you're even lucky, we'll say, oh, that's interesting. Can you tell us more info?
I always try and tell more info. I think if we're helping each other to learn, I learn, and other people will also learn if I can say why something is up there, and it's a good practice helping new people. Sometimes with some of these observations, they can actually sit there for months without being identified. And that's just the nature of it, particularly with Australian species generally. I've got one observation up for a whitefly. It is the only observation in the world on INATs. It may be common, but there's not many researchers, so most people don't know about it. I was fortunate that a researcher came along and said, yep, that's what it is, and good. So we've started the observations. The other reason is that observations, for the computer vision to work and give you a suggested observation, a suggested species name rather, it needs at least 100 observations to even begin to make a suggestion, which is why sometimes you get some really wild suggestions that don't make sense. It's because it doesn't have the data there to do it. I think you guys are a bit ahead of South Australia, but we've got so many that are just, we have less than 100 observations of. And on top of the fact that in Australia, we've got so many species that are not even named. So you do need to be prepared that not everything will necessarily get identified species level, but the data's there, and it means somebody can come along and use it to, yeah.
Does anyone have any other questions? Yeah, so normally when we do these presentations, we usually put the videos and the microphones off, but if you like to ask a question verbally, you can raise your virtual hand. So at the bottom, there's a little reaction tab, and if you click on that, you can raise your hand if you like to put on your video or audio.
Rosalie, you might have to stop sharing too.
Yeah, that's what I needed to do.
Oh, yeah. All right. Well, look, we're sort of a little bit over time, but that's all right. Yeah, look, thank you, Rosalie. Thank you, Greg, for being with us tonight. And thank you, everyone who attended. We hope you enjoyed the talk, and we hope this talk has inspired you to join up with Wild Orchid Watch and go out into the bush and collect data on our orchids. We'll be sending out an email this week with some resources, some links and resources, as well as a survey form for everyone to fill out. Won't take very long. It's only a 3-minute one. And yeah, that's it. Thanks everyone, and enjoy the evening.
Thank you.
Bye everyone.
And thank all for joining.
Bye.